involved in direct incompatibility between species of wild tomatoes. Okay, a prediction of the Dobzhansky-Muller model of speciation that was described by Alan Orr as a snowball is this idea that the number of Dobzhansky-Muller incompatibilities, or DMIs, should accumulate faster than linear with time. That happens because as two populations diverge and fix differences, then every new substitution has a larger and larger target for incompatibilities to arise. And there's a little bit of empirical evidence of the snowball effect, like this one from Leonie Moyle and Taknakasato, who in 2010 set out to ask about the number of QTLs that are involved in two fitness components, on the seed side, on the pollen side, of 3 pairs of species of wild tomatoes. So they used introgression lines and detected that the number of QTLs in the seed fitness component is actually following a quadratic that is consistent with the snowball effect. But on the pollen side, things seem a little bit more linear. So that begs the question of where those missing incompatibilities are. So where could those VMIs that we're not seeing are, and why is the snowball not happening? So the first possibility is that DMIs are actually of higher order, so not pairwise. Instead of having little a and little b being compatible, you need, like in this example, B, in this cartoon, B is incompatible with A and D, and you actually need the full hybrid that carries little a, little b, and little d before it manifests the sterility. A second alternative is that the known QTL, the ones that we are seeing, are actually involved in multiple Dobzhansky-Muller incompatibilities. That's a little bit counterintuitive because originally Alan Orr's model thinks about the probability of interaction between two loci as very, very low, right? So it is very unlikely that you find a single locus that is involved in multiple ones. But let me walk you through examples of how I think this would be working. So if you think about a network of genes or a set of genes that are acting together to produce a phenotype, like say controlled pollen sterility, then you can do a little cartoon like this one here, and each node is one locus, and then the red edges would represent an existing DMI. A new substitution could arise, like this guy in green here, and that gene could lead to that substitution could lead to an incompatibility with any of the other green lines over here. But it so happens that each one of those 3 loci is already part of a DMI, right? They are all connected. They all have their own little red line. So that the implication there is that if you were to count the number of QTLs here, there's a little bit of a disconnect with the number of loci that are involved in the Dobzhansky-Muller incompatibilities and the incompatibilities themselves. So you can think of so the higher probability of interaction would lead to a faster occurrence of this notion that I'm calling saturation. So I'm showing you 2 examples of 2 different cases or 2 different snowballs, rather, right? So on orange, we have a case in which genes have a very low probability of interacting. You can think about it as a very lowly connected network. And then the solid line represents how, like, the number of pairwise incompatibilities accumulates through time. And then the dotted line would represent the number of loci that are involved in these DMIs. And it kind of acts as a really good proxy, right? So if you counted QTLs in that case, it seems pretty good. On the other hand, I'm showing you this white case in which you can think about it as like a highly connected network or a set of genes that have very high probability of interacting. And that particular case has reached this saturation point. You've seen this pointer? Yeah. In which the QTL, the number of loci involved in DMI, is not anymore a good proxy for the number of DMIs that are accumulated. Right, so those are our alternatives, and with that in mind, to just trying to hunt down where the missing incompatibilities are, we set out to do a large number of crosses in which the parents are already introgression lines. So we have in this cartoon case, nil, or introgression line 1, carries an introgress region of Solanum heterorchides on the background of Solanum lycopersicum, which is domesticated tomato. And then nil 2 is the same thing, but for another portion of the genome. We're going to put those together and make them be nice and homozygous, and I'm going to call those DILs, or double introgression lines. I'm going to take those DILs and use the phenotypes of the nil parents and DILs and try to figure out if we can find extra or explanations for these missing incompatibilities. These are the number. This is a summary of what we did. We took 15 single introgression lines and we crossed them all pairwise combinations. And at the end of those trials, we ended up with 96 DILs. Now, this is just a number here, but this is a lot of work put together by Tacna Casado, who's not here. There's a lot of process in the greenhouse. And then from all of those, including the parents and actually the domesticated tomato, the background parent, we count seed counts for the seed sterility fitness component and the fraction of fertile pollen as an indicator of pollen sterility. Okay, and I'm going to show you a lot of panels like this one, so I'm going to walk you through it. We're going to, we have the fitness of the first parent and the fitness of the second parent, and with those we can come up with an expectation based on an additive model for the DIL. And then we can use a likelihood approach to compare whether the observed fitness of the DIL is explained by the sum of the additive or the additive components of the parents, or if it deviates from both. If there's a significant deviation from additive component or additive fitness, then I'm going to color the panel as this not so pretty pink on screen, and I will color it pink if the expectation is higher than the observed. That is, the nil is a worse hybrid than the sum of its parents. And I will color it green if the hybrid is better than the additive components of its parents, right? So it's over the expectation. Okay, and I'm going to put all these panels together. I'm going to show you in a single matrix. And this is kind of what we expect. So we have all the parents on the rows and columns. And each one of these parents are ordered by the size, the effect size. So all to the right and further down, they're the sterile nils and the non-sterile nils that we don't detect a significant effect with respect to the background parent. And so the expectation is that we will, if there's higher order DMIs, we'll see a lot of really crappy hybrids, or hybrids that are way worse than their parents, especially for parents that are non-sterile. So we'll see a lot of pink in this top area. On the other hand, if we have a lot of interactions among DMIs or QTLs that are involved in multiple DMIs, we'll probably see a lot of rescue effects, especially among sterile DMIs. Sterile nils, I'm sorry. Okay, so these are the results. On the left-hand side, we have the seed fitness component and the pollens on the right. And then the first thing that pops out is that there's absolutely no pink. There is no case in which the hybrid was significantly worse than the additive components of the parents. The second thing that is striking is that seems to be some sort of accumulation of green area over here. That means there's a lot of hybrid rescue, especially in the pollen. That doesn't seem to be the case for seed. Right? So basically the QTL that we have for pollen seem to be interacting a lot more among themselves. There's a lot more rescue there. I can show you those same results in a network way, and it looks kind of like this. So the orange nodes are the sterile parents, and the blue nodes are the non-sterile parents. And you can see that if each edge is a significant interaction or significant epistatic effect between these nils, then the pollen side seems a lot more connected than the seed side. So it seems like even though it's a little bit hand-wavy and it's not really a direct test, the seed patterns of interactions might be consistent with the fact that we actually see. So we're seeing the snowball effect effect in the seed side. So the number of QTLs that we're seeing in seed are snowballing nice and pretty over here. It's kind of consistent with a network that is not saturated, or that has been, or that is basically very lowly connected. Oops, sorry. And on the pollen side, if we're seeing, when we count the number of QTLs, a linear effect, it might be because those QTLs are not anymore, not a very good proxy for the number of incompatibilities anymore, right? And that could also mean that the network of genes that produces the pollen sterility has either fewer genes or highly connected genes, or have other sort of differential selection stuff going on that is quite controversial and I won't get into. So in conclusion, I think I've shown you that interactions affecting hybrid sterility are very common. We found a lot of them in our classes, and all of them are antagonistic. That means that the direction of the epistatic effect is against the direction of the hybrid, of the contribution of the parents, right? Because all of the ones that we saw were hybrid, led to hybrid sterility, then hybrid rescue means hybrid antagonistic selection. Or, sorry, antagonistic epistatic effects. Right. And then I show you that the patterns are exaggerated for pollen sterility phenotypes. That is, most of the antagonistic interactions that I showed you are accumulated in the sterile pollen lines, and that those interactions could be a good hint that the sterility QTLs that we're seeing in pollen are actually part of what I'm calling a saturated network that might explain the patterns or the lack of snowballs. With that, I would like to acknowledge the work of Chris Muir and Sarah Josway. Chris did a lot of statistical work for this before, and Sarah did a lot of process with TAC and Moyle and Han labs in the university. There's plenty of time.
Do I have a reason for it?
How can I speculate about it? Yeah, I think that, I mean, it's a little bit, it's very humbling, I guess. I think pollen is weird. It is very likely that there are very few genes contributing to just pollen germinating or pollen actually persisting as a whole, right? So many of these times pollen sterility, that fraction of fertile pollens, the sterile pollen actually is just a husk. It's not, like nothing happens. So you can see how, you can think about it as that particular phenotype is very close to like some sort of master regulator or something like that. It's very high up in some sort of pathway, and then maybe fewer genes are being involved in.